Hey everyone, welcome back to Windows 8 Studies. I hope you all are doing well. So we started discussing about immunology, where we first learned about what is immunity, what are the different types of immune systems that are found in humans. Then we also learned about the cells, the hematopoietic stem cells, which are going to give rise to the all of the mature blood cells. So we learned about how the generation of blood cells from hematopoietic stem cells occur. So we learned about hematopoiesis, the process of differentiation and maturation of hematopoietic stem cells into natural blood cells. So we learned about that. So there we discussed about the different lineages that are found in the blood cells. So we discussed about the immune cells, the leukocytes. They are of two types, the granulocytes as well as the agranulocytes, depending upon whether the cytoplasm of those leukocytes, means the white blood cells, whether they have the granules or not. So we started discussing about granulocytes. So in last lecture, we learned about neutrophils, the major type of WBCs or major type of granulocytes that are found in our blood. So we learned about neutrophils in last class. Today we are going to discuss about the rest of the leukocytes, the eosinophils and the basophils. So we learned about the granulocytes. So granulocytes are the cells which have granules in the cytoplasm. So granulocytes are categorized into three different types based on their morphology or the staining procedure or the results of staining procedure. So the granulocytes, if they can be, they can be stained by both acidic dye and the basic dye. They are referred as neutrophils. If they are stained by the acidic dyes, they are referred as eosinophils. All of them, again, not just staining results are different, but the morphology of those cells are also different. The number of lobes that are found in the nucleus. So, for example, the neutrophils are multilobed. On the contrary, the eosinophils and basophils, both of them, they are bilobed. So, the third type of the granulocytes are the cells which can be stained by the basic dyes. They are referred as basophils. So those three types of cells, those three types of granulocytes, they are found in the blood. But besides them, there is a category of the granulocytes which is found in the tissue, and that cell type is known as mast cells, which results into allergic reactions. So we will be discussing about mast cells in next lecture, in a separate lecture. Today's point of discussion is eosinophils and the basophils. We have already discussed about neutrophils. So today we are going to discuss about eosinophils and basophils in detail. So let's start discussing about eosinophils. So as I already told you, eosinophils are the granulocytes which can be stained by acidic dyes. So those can be stained by acidic dyes such as eosine, okay, and those acidic dyes are negatively charged, right? So if these types of cells, they can be dyed by the negatively charged dyes. This means that whatever content this particular cell has, it should be positively charged. This means that eosinophil has basic proteins. Okay, so the cytoplasm or the granules of eosinophil has basic proteins, and that is why to those basic proteins the acidic dyes can bind, and it gives it when viewed under the microscope we can see a pinkish red or a reddish pink type of cells, and those are eosinophils. So eosinophils will be visualized as the reddish pink cells which has bilobed nucleus. Okay, so if you view them under microscope, you will see something like this. Okay, so there will be a cell which will have the bilobed Nucleus. So this is the nucleus, which has two lobes. So the nucleus will be something like this. So this is the bilobed nucleus. And it will also have granules. So this is going to have multiple granules, and that is why it is a part of granulocyte. It is categorized into granulocytes. So when those granulocytes zoomed, if we zoom in those granulocytes, so what we can see, those granulocytes, they have a lipid membrane. Okay, so they are made up of a lipid membrane. Okay, so inside those granulocytes, in this lipid-bound granules, so these are the granules which are lipid bound. So this is a lipid bilayer, the pink in color, and it has a crystalloid core. Okay, so this is the crystalloid core of the granules, which will have the basic proteins. So the crystalloid core is going to have basic protein, whereas the other part is known as matrix. So this orange colored, the one I'm shading with orange color, this is the matrix of the granule. So this is the structure of granule. There exists the matrix and there exists a crystalloid core. So the matrix and crystalloid core, both of them, they have different types of proteins. So the crystalloid core of the granules of the eosinophils, it is made up of majorly basic protein, which is known as MBP. So MBP is the major basic protein, which is found in the crystalloid core of the granules of the eosinophils. Whereas the matrix of the granules of those eosinophils, it has different proteins such as ECP, EDN, and EPO. So we will discuss about all of those different types of proteins found in the granules of eosinophils one by one. So the MBP, okay, the first one is the major basic protein. So it is quite clear from the name that this protein is basic in nature, means it is a positively charged proteins. So this is highly cationic protein, means it is a positively charged protein. And due to this, the negatively charged dyes like eosine can bind to the eosinophils, giving them a pinkish color. Okay, so though this MBP has two isoforms. Okay, the two isoforms are named as MBP1 and MBP2. So there are two isoforms in the crystalloid core of eosinophils, MBP1 and MBP2. So those the what does major basic protein do? They work as a toxin for helminths. So the eosinophil, so as discussed before, neutrophils, they are the phagocytic in nature. Eosinophils, they are also phagocytic in nature, but that is not their primary function. Neutrophils are more aggressive compared to eosinophils. Both of them show phagocytosis, but eosinophils, they are majorly known for having their effect against the worms, against the parasites such as ascaris, such as Taenia solium. So whenever there is parasitic infection, the cells which are going to which are going to attack that parasite and will help in protecting the body against that parasite are the eosinophils. Basophils, they also help in parasitic infections, but the major type of cells which are going to help in parasitic infections are eosinophils, because they have those basic proteins, major basic proteins, and those major basic proteins, they work as helminthotoxin. Okay, helminthotoxin means they are a potent toxin against the helminths, against the parasites, against the worms. So they are going to, what will they do? So those major basic proteins, they are positively charged. So they are going to bind to the negatively charged phospholipids present on the membrane of the helminths. So the parasitic membrane. Okay, so this parasitic membrane will have negatively charged phospholipids, right? So to this negatively charged phospholipids, this lipids phospholipids. To those phospholipids, the MBP is going to bind, and due to the binding of MBP to those phospholipids, the parasitic membrane of will be disrupted. So this will result. This is helminth toxin because it results into disruption of parasitic membrane. Right, and this is how it kills the parasites, the very large parasites such as Ascaris, Taenia sodium. So this is very effective against them. So the other way that by which basic protein works is that it is not just helminthotoxin, but it also it also activates other immune cells such as basophils, such as mast cells. Okay, so the activation of mast cells or basophils by the major basic protein of eosinophils, it will result into histamine release. Okay, so the basophils as well as mast cell, both of them have histamines in them. Okay, so this will result in histamine release, and this histamine, this is actually going to result into inflammation. Okay, we already know that histamine is an inflammatory chemical, so it is going to result into inflammation, but alongside inflammation, it will also result into bronchoconstriction, or we can also call it bronchospasm. So this bronchoconstriction or bronchospasm, it refers to the narrowing of airways due to the contraction of smooth muscles of those bronchioles. So bronchioles, they have smooth muscles in them. So those smooth muscle will contract due to histamine, and that will create, that will result into narrowing of those airways. So why does during inflammation bronchospasm is necessary? So this is a way, this is a protective mechanism of our body to ensure that if the airways they are narrowed down, this will ensure the less entry. So this will prevent, or this will limit the entry of allergens, entry of pathogens through the airways. So this is a kind of exaggerated protective mechanism that is exhibited by our body. So it is going to this. So bronchospasm is usually done due to to ensure that the entry of the pathogen will be limited. So it limits the entry of allergens or entry of pathogens. Okay, so this the histamine release will result into bronchospasm, which will also result into sneezing and coughing. Okay, and due to sneezing and coughing, why does this sneezing and coughing due to the inflammatory responses is important? Because when we sneeze and cough, we are spilling out the pathogens, we are spilling out the allergens through the airways. So that ensures that, again kind of ensures that whatever allergens are being they're entered in the body, they get they get thrown out. So this will result into throwing the allergens out of the body. So this is kind of the protective mechanisms that our body ensures against the pathogens, okay, against the allergens. So the major basic protein. So what does it do? It is a helminth toxin, and it also activates mast cells. These are the two jobs that are performed by the major basic protein. So besides the major basic protein, eosinophils has other other proteins as well that we discussed, the ECP, EDN, and EPO, which are present in the matrix of the granules. So the other proteins, okay, the ECP is the eosinophil cationic protein. Okay, so it is also a positively charged protein. So this is eosinophil cationic protein. So this cationic protein, it is a member of RNase A family. Okay, so it is a member of RNase A superfamily. So this is a ribonuclease. Means it is going to cleave the RNA. So it will be very beneficial against the viruses which have RNA as their genetic material. So this is how the ECP has antiviral property, effective against the viruses with RNA as their genetic material. And besides antiviral property, it also has antiparasitic and antibacterial properties. Because this is not this RNAs is not just going to cleave off the cleave of the RNA of the viruses, but it also disrupts the membrane of parasites. And when it binds, so what does it do? The ECP, it binds to the membrane of the parasites or membrane of bacteria. So it binds to their membrane, and the binding of ECP to those membrane will create pores in the membrane, and that will end up killing the pathogen, killing the bacteria or the parasite. So that is why the ECP, eosinophil cationic protein, also has antiparasitic and antibacterial properties. Okay, the other protein that is found in the matrix of the eosinophil granules is the EDN. Okay, which is eosinophil derived neurotoxin. So this protein has neurotoxic properties. So it also belongs to RNase A family. So this is also a ribonuclease which is going to be effective against the against the pathogens, but this is less cytotoxic. So compared to, the killing capacity of EDN is it is not that effective, so, like eosinophil, but the RNA's activity and the whatever RNA's activity means, the RNA cleaving capacity EDN has, it is much stronger than ECP. So this is less cytotoxic but effective antiviral protein of the eosinophil. So this is less cytotoxic but strong antiviral protein. Okay. So besides those ECP and EDN, the fourth protein that is found in the eosinophil matrix, matrix of granules, is the EPO, which is eosinophil peroxidase. So as the name suggests, it is a peroxidase, means it is going to convert the hydrogen peroxide into the H bleach or HOBr. So it is going to convert the hydrogen peroxide into the bleach, which is going to result into killing of pathogens. Okay. So we can say that eosinophil, they are not a fan of production of ROS, but there exists a protein called eosinophil peroxidase, which is going to convert the hydrogen peroxide into bleach, which is going to result into the killing of pathogens. So this also, besides the besides the bleach formation, the EPO, this peroxidase, is also important in immunomodulation. So it modulates the immune responses by triggering the cytokine release from the epithelial cells. So it will trigger the cytokine release from epithelial cells, okay, which will result into the immunomodulation. It is going to activate other immune cells. Okay, so besides those proteins, eosinophil, they also secrete cytokines so that they can alert other cells such as basophils or neutrophils even. Okay, so these are the major proteins that are secreted by the eosinophils. Okay, so let's now discuss about basophils. With this, I'm done with eosinophils. We have discussed about eosinophil, the composition of eosinophil granules. We have discussed about the properties of eosinophils granules. So eosinophil, they are reddish pink in color, but the basophils, on the contrary, which are being dyed with the basic dyes which are positively charged. Okay, so those basic dyes are positively charged, such as methylene blue. Okay, so it will result into bluish purple cells. Okay, and those bluish purple cells, which has multi-lobed, bilobed nucleus, which somewhat looks like S shape, are known as basophils. So basophils, they are also granulocytes, which are, which are bilobed, just like eosinophils, but they can be dyed with the basic dyes such as methylene blue or the purple dyes, and that is why it results into a bluish purple cell. So basophils looks like this, like this. Okay, so this is the basophil cell, which will have a bilobe nucleus. Okay, so this is how the nucleus looks like of the basophils. So it's not really, we can't really say that it is perfect S shape, but it somewhat looks like S shape. Okay, so this is a bilobe nucleus. Of the basophil, it is going to have granules. Okay, so basophils have large granules in it. Okay, quite distinct granules. It doesn't have small granules, and so those granules, they have histamine and heparin. So they are going to release the inflammatory mediators, the inflammatory chemicals such as histamine and heparins. We have discussed this before also. So let's discuss about the properties that basophils have. So basophils have surface markers which can bind to the IgE antibody. So there are 5 types of antibodies in the humans, and I haven't discussed antibodies yet, but so if you don't know about antibodies for now, just remember that IgE antibody can bind to the surface markers, means the proteins which are expressed over the basophil plasma membrane. So the surface of basophils have proteins, okay, have receptors, proteins which are going to act as receptors for IgA antibody. So those receptors that are present on the basophils, those are the surface markers. Means if we have an antibody against those receptor and that antibody is binding to certain cell, then we can for sure say that this is the basophil. Okay, so those receptors are Fc epsilon R1. Okay, so remember the name Fc epsilon R1 are the surface markers for basophils, and to those receptor binds the IgE antibody. So this is IgE antibody, so it can bind to the basophil with the help of those surface markers. So the binding of this IgE antibody to this surface marker will result into release of histamine. So this is going to release the histamine in the environment, in the microenvironment, and that is going to result into the inflammation. We have already discussed that histamines are the they are the vasoactive amines, means they are going to do the vasodilation. So they are vasoactive amines, means they have NH2 group. Okay, and they are vasoactive molecules, means they are going to result into changing the vasculature by dilating it. So they do the vasodilation. So they are vasodilated. In the lecture where we discussed about neutrophils, we discussed that the release of histamine is very important because it is going to result into vasodilation, and due to vasodilation, now the the cells in the blood vessels, they can release this, they can escape. So due to the vasodilation, the histamine mediated vasodilation will result into extravasation of the immune cells. Extravasation means the cells which are present in the blood vessels, the immune cells, they are going to, they are going to move from the blood vessel, from the blood to the interstitial tissue without rupturing that blood vessel. This is also known as diapedesis. If shown by if the immune cells are the ones which are showing this phenomena, it is called diapedesis. And this is also shown by tumor cells, means from, this is the blood vessel, and in the blood vessels there are the immune cells. If the immune cells are moving from blood vessel to the interstitial tissue, that is known as diapedesis. Okay, and it is also known as extravasation in the case of tumor cells. So this diapedesis, this is caused due to the vasodilation, which is mediated by histamine, and this histamine is released by basophils. By the way, histamine is also released by mast cells. So histamine can be released by basophils as well as mast cell, which is an inflammatory chemical. So this chemical is going to cause the inflammation, and it is going to result into allergic reactions. So what does it do? It causes allergic reactions. It causes smooth muscle contraction. It also results into itching, and that is why when the histamine is overdosed, whenever there is excessive histamine release, excessive activation of those immune cells which are going to release histamine, it will result into bronchoconstriction, and it can also result into asthma. Okay, this clinical condition can also be caused due to the release of histamine, and that is why in those cases, whenever in the allergic reactions, whenever a person is having allergic reaction, we give them antihistamines such as cetirizine. Okay, the cetirizine, it binds to H1 receptor. It is an antihistamine. So that is how we control the allergic reaction and giving relief to that patient. Okay, so histamine is released by basophils, but alongside histamine, the other molecule which is there in the granules of the basophils is the heparin. Okay, so heparin is an anticoagulant. Okay, heparin is an anticoagulant, glycosamine glycan. So it is a glycosamine glycan molecule, which has anticoagulant property. So why does heparin, which is an anticoagulant, is released by the basophils? Because it is anticoagulant, means it has capacity to prevent the clot formation. Coagulation means clot formation. So heparin release will, result into prevention of clot formation. And why is it necessary? Because whenever the inflammation is there, the clot is going to form in the pus pocket. Okay, and that is going to create the pus pocket, and it is necessary to, to localize that infected, infected area, to localize that bacteria or localize that pathogen in that infected area. But anticoagulation allow in the inflamed tissue is very important, so that the blood flow will be maintained in that inflamed tissue. If the blood flow is maintained in that area, in that particular tissue, it will result into, the ease of entry of the immune cells, and that is going to result into quick immune response. And that is why anticoagulant release, the heparin release, is very important, because whenever in the inflamed tissue, the blood flow will be maintained. So, blood is flowing in that inflamed tissue, so anticoagulation will not let the blood clot there, and it will result into only the pus pocket, and it is going to result into the blood flow, the maintained blood flow in that particular tissue. So heparin anticoagulant, it maintains blood flow in inflamed tissue, to the inflamed tissue. Okay, so besides heparin and histamine, basophil, they also release cytokines. So even eosinophils, basophils, both of them, they are going to release cytokines so that the cytokines can recruit other immune cells. Okay, remember the neutrophils, they also work if by due to the cytokines. So neutrophils have the, have receptors for which are going to receive the cytokines. Eosinophils can also be recruited due to cytokines, and basophil can also be recruited due to cytokines. So cytokines are the chemical mediators which are going to recruit those, which are going to activate those immune cells and evoking the immune response. So these are the major proteins that are secreted by the eosinophils and basophils upon degranulation, and this is how they help in the immune responses in the killing of the pathogen. Okay, so let's summarize the whole lecture once. So today we learned about eosinophils and basophils, which are a type of granulocytes. Means in their cytoplasm there will be granules containing multiple proteins which are helpful for killing of the pathogens. So eosinophils, they are the phagocytic cells, and they also release some proteins which are going to kill the large parasites such as helminths, such as the worms, the ascaris, the tinea solium. So it is helpful in the worm killing. Okay, so neutrophils, they can work by the ROS production against any type of pathogen. Eosinophil, basically they have majorly, they target the parasites, the large parasites such as ascaris and the other worms. Okay, whereas basophils, they are non-phagocytic. Okay, so phagocytosis can be exhibited by neutrophils and eosinophils, but the primary function of eosinophils is not phagocytosis. Eosinophils are the acidic, they can be dyed with the acidic dyes, and basophils, they have basophilic proteins, and that is why they can be dyed with the basic dyes. The eosinophils, they look reddish pink under microscope, while basophils, they are the bluish purple cells. So eosinophils have the granules which are going to have a crystalloid core as well as the matrix, whereas the basophils have simple granules, but they are very large compared to the eosinophils. So both of them, they are bilobed, they have bilobed nucleus, while the neutrophils, they have multi-lobed nucleus, unlike those. Okay, so the bilobe nucleus in the basophils, it is S-shaped. The eosinophils, they have four major proteins in their granules. In the crystalloid core is the major basic protein, and in the matrix are the ECP, EDN, and EPO proteins. Okay, so all of them, they are going to result into the elimination of parasites. So major basic protein eliminates parasite by binding to the phospholipids, the negatively charged phospholipids over the parasitic membrane, and it kills the parasite. That is why it is a helminthotoxin. So MBP is a helminthotoxin. ECP can also, it can also bind to the parasitic membrane, and it results into the killing of pathogen. Okay, so killing of the parasite, and it also has antiviral properties. So both of them, the ECP and EDN, both of them, they belong to the RNase A family. Okay, while the EPO is a peroxidase. So those four major proteins, they are there in the granules of the eosinophil, and basophils, they have histamine and heparin in their granules. Both of the eosinophils and basophils can release cytokines to do the immunomodulation, to recruit the other immune cells. So if you have to define those cells, the neutrophils, basophils and eosinophils in one sentence, how will you define? So neutrophils are the granulocytes which mediate the killing of pathogen by ROS production, the reactive oxygen species production. They are very aggressive in nature, and they are also phagocytic in nature. The eosinophils are the granulocytes which kills the parasites, the big parasites, by having the basic proteins, the major basic proteins, and they are the bilobed in nature. The basophils are the ones which are going to mediate the histamine release, which is actually an inflammatory mediator resulting into vasodilation. So this will also help into the inflammatory response, and it also helps in maintaining the anticoagulation in that particular area, in the inflamed tissue. So with this, I'm done with the granulocytes, which are present in the blood. In next lecture, we'll discuss about mast cells, which are also a type of granulocytes, which have big, large granules in their cytoplasm, and they are going to mediate the pathogen killing in the tissues, in the infected tissue. So those are the tissue cells, tissue granulocytes, while those three are found in the blood. So they are released in the tissue from the blood without rupturing the blood vessels, while the mast cells, they are already present in the tissues. So I conclude the lecture here. In next lecture, we'll discuss mast cell. Till then, see you. Bye bye.